Hello everyone. Today we will discuss about the metabolism of tryptophan. This tryptophan is a heterocyclic amino acid. Chemically, it is alpha amino beta 3 indole propionic acid. This tryptophan is an essential amino acid. Coming to the metabolism of tryptophan, major pathway of this tryptophan catabolism is known as Kynurenine Anthranilate Pathway. Kynurenine Anthranilate Pathway. This tryptophan, it is both glucogenic and ketogenic amino acid. It is both glucogenic and ketogenic. Because in this pathway, this tryptophan is finally converted to glutaric acid. Tryptophan, it is converted to glutaric acid. Then this glutaric acid form acetoacetyl-CoA. From this acetoacetyl-CoA, two acetyl-CoA molecules are formed. So it is ketogenic. Then it also produces alanine. This alanine on transamination can form pyruvic acid. So this tryptophan is glucogenic. So tryptophan is both glucogenic and ketogenic. So let's see the pathway that is known as Kynurenine Anthranilate Pathway. First step is conversion of tryptophan to N-formyl kynurenine. Tryptophan to N-formyl kynurenine. This is by the enzyme tryptophan pyrrolase. Tryptophan pyrrolase. This enzyme is an iron porphyrin metalloprotein. It is an inducible enzyme in liver. Its synthesis is increased by glucocorticoids and also by tryptophan. It is increased by glucocorticoids and also by tryptophan. Then this enzyme is feedback inhibited by NADPH. So this is the first enzyme, that is tryptophan pyrrolase. This is the first step. Then the second step, it is a conversion of this N-formyl kynurenine to kynurenine by the enzyme kynurenine formylase. Kynurenine formylase. One molecule of water is added and here formate is released and it forms kynurenine. This is the second step. That is the conversion of this N-formyl kynurenine to kynurenine by the enzyme kynurenine formylase. Third step is catalyzed by the enzyme kynurenine hydroxylase. Kynurenine hydroxylase. This step requires NADPH plus H plus. And this kynurenine, it is converted into 3-hydroxykynurenine. 3-hydroxykynurenine. This is the third step. This kynurenine is converted to 3-hydroxykynurenine by the enzyme kynurenine hydroxylase. Here it requires NADPH. Then next step is conversion of this 3-hydroxykynurenine to 3-hydroxyanthranilate. 3-hydroxyanthranilate. 3-hydroxykynurenine is converted to 3-hydroxyanthranilate. Here the enzyme is kynureninase. Kynureninase. This is the fourth step. This enzyme requires pyridoxal phosphate. In B6, this vitamin B6 deficiency, this 3-hydroxykynurenine cannot be converted to 3-hydroxyanthranilate. As a result, this kynurenine and 3-hydroxykynurenine accumulate in extrahepatic tissues and they are converted to xanthurenic acid. This kynurenine and this 3-hydroxykynurenine cannot be kynurenine cannot be converted to 3-hydroxyanthranilate. As a result, the kynurenine and the 3-hydroxykynurenine accumulate and it is converted to xanthurenic acid in extrahepatic tissues. Xanthurenic acid. This xanthurenic acid it is excreted in urine. So this xanthurenic acid excretion in urine is an index of this B6 deficiency. Its excretion is an index of B6 deficiency. Here this 3-hydroxykynurenine can form alanine. 3-hydroxykynurenine it can form alanine and by transamination this alanine can be converted to pyruvate. So this tryptophan is glucogenic. The next step is the conversion of this 3-hydroxyanthranilate to 2-acrylyl 3-amino fumarate. 3-hydroxyanthranilate it is converted to 2-acrylyl 3-amino fumarate. This is the next step. This is by the enzyme 3-hydroxyanthranilate oxidase. By the enzyme 3-hydroxyanthranilate oxidase, this 3-hydroxyanthranilate it is converted to 2-acrylyl 3-amino fumarate. Then this 2-acrylyl 3-amino fumarate it can be converted to quinolinic acid. Quinolinic acid, or it can be converted to by decarboxylation it can be converted to 2-amino cis-muconate semialdehyde. So this 2-acrylyl 3-amino fumarate it can be converted to quinolinic acid or 2-amino cis-muconate semialdehyde. This quinolinic acid then forms nicotinic acid or niacin. This 2-amino 6-muconate semialdehyde then converted to oxalocrotonate. Here NADH plus H plus NAD plus NADH plus H plus NAD plus, here ammonia is released. And this oxalocrotonate it can be converted to alpha ketoadipic acid or alpha ketoadipate. Here NADPH plus H plus NADP plus alpha ketoadipate. This alpha ketoadipate then converted to glutaryl CoA. It is converted to glutaryl CoA. Glutaryl CoA then forms acetoacetyl CoA. From acetoacetyl CoA, 2 molecules of acetyl CoA are formed. So this tryptophan is ketogenic. So it forms alanine then to pyruvic acid, so it is glucogenic. Then here it forms acetoacetyl CoA to 2-acetyl CoA, so it is ketogenic. That's why it is known as both glucogenic and ketogenic amino acid. This is the major pathway that is known as Kynurenine Anthranilate Pathway. So coming to the metabolic roles of this tryptophan. This tryptophan can form nicotinic acid or niacin. Then it gives rise to the formation of melatonin and serotonin. Then on transamination, this tryptophan forms indole pyruvic acid. Tryptophan on transamination, it forms indole pyruvic acid. Transamination. This indole pyruvic acid By reduction, it is converted to indole lactic acid. Indole lactic acid. Then by decarboxylation, this indole pyruvic acid forms indole acetic acid. Indole acetic acid. So these are the metabolic roles of this tryptophan. That is, it can form nicotinic acid, give rise to serotonin and melatonin. Then on transamination, tryptophan produces indole pyruvic acid. Then on reduction, indole pyruvic acid is converted to indole lactic acid. And by decarboxylation, it is converted to indole acetic acid. So this is known as, this first pathway is known as Kynurenine Anthranilate Pathway. Then another pathway of this tryptophan metabolism is serotonin pathway. Serotonin pathway. This serotonin is chemically 5-hydroxytryptamine. 5-hydroxytryptamine. This serotonin is a vasoconstrictor. Vasoconstrictor. It is present in the blood and is produced in tissues like gastric mucosa, intestine, then brain and mast cells. And it is stored in platelets. It is stored in platelets. Then this serotonin, it is produced by serotonin producing cells. Produced by serotonin producing cells. These serotonin producing cells can take up silver staining. They can take up silver staining. So these are called as Argentafin cells. Argentafin cells. So these serotonin producing cells can take up silver staining. So these are known as Argentafin cells. So let's see the synthesis of serotonin. First step is this tryptophan is first hydroxylated by the enzyme hydroxylase and it forms 5-hydroxytryptophan. Tryptophan by the enzyme hydroxylase, it forms 5-hydroxytryptophan. This reaction is analogous to conversion of this phenylalanine to tyrosine. In the next step, this 5-hydroxytryptophan is decarboxylated by the enzyme 5-hydroxytryptophan decarboxylase. Next step is decarboxylation. By the enzyme decarboxylase, carbon dioxide is released. It requires PLP and it forms 5-hydroxytryptamine. 5-hydroxytryptamine. It is otherwise known as serotonin. This is the formation of this serotonin. This enzyme decarboxylase, 5-hydroxytryptophan decarboxylase is present in kidney, liver and in stomach. So next step is the conversion of this serotonin, this 5-hydroxytryptamine, that is serotonin, to 5-hydroxyindole acetic acid. 5-hydroxyindole acetic acid. This 5-hydroxyindole acetic acid that will be excreted in urine. Then the serotonin, it can form melatonin. Melatonin means it is a hormone of pineal body and peripheral nerves. It is a hormone of this pineal body and peripheral nerves. This melatonin, it is synthesized from this serotonin. So in the formation of this melatonin, first step is this acetylation of this serotonin by the enzyme acetylase. By the enzyme acetylase, it is converted to N-acetyl. The serotonin is converted to N-acetyl serotonin. Here, acetyl-CoA acts as acetyl donor. Acetyl-CoA, it acts as acetyl donor and it forms N-acetyl serotonin. Then next step is methylation of this N-acetyl serotonin. Methylation of N-acetyl serotonin and it forms melatonin. Here S-adenosyl methionine, that is active methionine that acts as the methyl donor, and it is converted into S-adenosyl. This S-adenosyl methionine, it transfer methyl group and it forms S-adenosyl homocysteine. Here the enzyme is N-acetyl serotonin methylferrase. N-acetyl serotonin methylferrase is the enzyme. So this step is methylation of this N-acetyl serotonin. Here S-adenosyl methionine, that is this active methionine, acts as the methyl donor. Here the enzyme is N-acetyl serotonin methylferrase and it forms melatonin. That is the synthesis of this melatonin from serotonin. So this is the metabolism of tryptophan. Coming to the inherited disorders of this tryptophan metabolism. A malignant tumor of this serotonin producing cells, it is known as carcinoids or Argentaffinoma. So malignant tumor of this serotonin producing cells is known as carcinoids or Argentaffinoma, and the clinical features are known as carcinoid syndrome. Symptoms are mainly due to, here the symptoms are mainly due to presence of excessive amount of this tryptophan, excessive amount of serotonin produced by malignant cells. Symptoms are mainly due to presence of excessive amount of this serotonin produced by malignant cells. So this is one of the inherited disorders of this tryptophan metabolism. Next one is Hartnup disease. Hartnup disease. This is a hereditary disorder associated with the defective tryptophan metabolism. Probably impaired formation of transport proteins. Of this tryptophan and other neutral amino acids in brain, intestinal mucosa and in renal tubular epithelial cells. So there will be impaired formation of transport proteins for tryptophan and neutral amino acids in brain, then in renal tubular epithelial cells and in intestinal mucosa. There will be defective transport of this tryptophan and other neutral amino acids. Defective transport. These are characterized by mental retardation, then neurological symptoms and pellagra-like skin rash. Mental retardation, neurological symptoms and pellagra-like skin rash. Then there will be decreased synthesis of serotonin and this nicotinic acid. Decreased synthesis of serotonin and niacin or nicotinic acid. This plasma level of tryptophan and other neutral amino acids, this will be reduced. Tryptophan and neutral amino acid. Plasma level of tryptophan and neutral amino acids are reduced. Then tryptophan is excreted in urine and in feces. Then urine also shows increased amount of indole acetic acid. These are the inherited disorders of tryptophan metabolism. So this is about tryptophan metabolism. This is today's topic. Thank you for watching.